Hello everyone, my name is Andrew Hunt. I've been interning at the Gutman Laboratory, and I'm going to talk to you guys a little bit about immune regulation of NF1 brain tumor growth and the process by which I've done this research. So first, just a little bit about me. I'm a senior at St. Louis U High, so roll bills. I'm involved in a variety of other things other than research at my school, and my parents went to Wash U, so I have a lot of connections with the university. So firstly, when we're talking about NF1 brain tumors, what is neurofibromatosis type 1? Essentially, it's a cancer predisposition disorder. It's usually genetic, but it can also be spontaneous. And what it means is that people with neurofibromatosis type 1 do not have the NF1 gene, which is a tumor suppressor. So without that, then that can cause tumors to grow. But there's no universal phenotype to define this. It's clinically heterogeneous. But there's a few common indicators, and one is neurofibromas, which a bunch of other labs study. Those are really, really common, and usually what people think of when you're talking about a patient with neurofibromatosis. So those exist in nerves under the skin and may result for 11% of NF1 patients in malignant peripheral nerve sheath tumors. And then the second common indicator of neurofibromatosis type 1 is optic nerve gliomas, and this is the focus of my lab, the Gutman Laboratory. So they're usually abbreviated ONGs or OPGs for optic pathway gliomas, and they're generally benign, but they're pretty common. So 15 to 20% of NF1 patients will have these, and obviously they're in the optic nerve, so that's between the eye and the brain. So they can result in proptosis, but they can also result in blindness. So they're a really big issue, and they're particularly found in children. And my lab has an emphasis on low-grade gliomas, or LGGs, which are slower growing. So essentially, studying OPGs, my lab has this process to go about the study of these, and that's to utilize genetically engineered mouse models to firstly investigate various pathways by which these tumors grow or these tumors form. That's tumorigenesis. And then secondly, to pinpoint potential therapies at some point upstream or downstream along this pathway to eventually inhibit tumor growth. So at the bottom of the screen here, you can see a few examples of research focus that people in my lab have done in the past or are currently working on. So there's various things within the tumor microenvironment or beyond and the organism as a whole that researchers in my lab have identified as contributors to the growth of optic nerve gliomas. So how do we go about investigating these pathways and identifying therapies along these pathways? I made this little graphic, which kind of lays it out pretty well of how the whole process goes in my lab, and surely it's something that also applies to many labs across this field. So it all starts with a research question, and you're asking why is this? Why do you notice this one thing happening, contributing to the tumor growth? And then you're making a hypothesis, and after you're making a hypothesis, you're testing, is that true? So you're looking at a mouse and you're making a model and you're treating the mouse in a certain way. And then after the mouse has lived its life and you have your control mice living their life and it's been treated and everything, then you're extracting usually the brain, the optic nerve, the eyes, and you're staining them for certain types of cells like tumor cells, microglia, etc. So then you can eventually analyze those, image them, and say, is my hypothesis correct? Is this accurate? And then you get into a stage of you're writing all this down, you're compiling all your results, and then often you're getting feedback from peers of what new treatments you should try, what new models you should explore before you finally go to the publishing stage. So that's a little bit about how the lab functions as a whole, and there's many different pathways that many different research focuses that all follow that same path. And then I would like to talk about my particular research, which has been with Dr. Rasha Barakat, who is a researcher in my lab. And I've been doing research under her research question, which is how do various immunotherapies affect tumor growth? So that in that case, we're talking about treatments that utilize the natural immune system with the goal of regulating optic gliomas. So we're particularly focusing on T cells, which are immune cells in the brain that they're focusing on the ones that are CD8 plus exhausted, which means they're not performing their proper function, and instead they're actually contributing to the growth of optic pathway gliomas. So we're focusing on these exhausted T cells and how we can minimize their contributions towards tumor growth. So there's two distinct different ways that they contribute to tumor growth. One is through the cytokine TGF-beta, and then also through CCL4. So to address those two different issues, I've explored two primary treatments. And the first is anti-TGF-beta, which, as the name says, it's targeted to reduce TGF-beta levels, which is, as I said, a cytokine that's involved in the immune cell, T cell mediated tumor growth. And then secondly is anti-CTLA4. And that actually attempts the de-exhaustion of these CD8 T cells so they can go back to their proper function, become unexhausted. And like I said, if you unexhaust those CD8 T cells, then you're inhibiting the production of the cytokine CCL4, which results in tumor growth again. So those are two potential things we've been exploring in this current phase of maybe these can contribute to reducing the tumor growth. So for this particular research, the process by which these the mice have been treated is that they live for 12 weeks, and then at 12 weeks, they begin being treated for 4 weeks, and then they're harvested at 16 weeks. So that means we're extracting the eyes, the brain, the optic nerve. And this is being done for the genetically engineered mice as well as, for treated mice, but there's also some mice that are given a placebo, which you'll see over here. So for both the CTLA4 group and the TGF-beta group, we have treated mice, so anti-CTLA4, anti-TGF-beta, and you can see that on the left, and you have IgG control, which means they're given some sort of placebo. That means they're not actually receiving the anti-CTLA4 or anti-TGF-beta treatment. And then under both of those two groups, you've treated, untreated, we're looking at the optic nerve and the eye particularly. And there's 3 stainings for the optic nerve, which is Ki67, IBA1, and CD8+. And then for the eye, there's SMI32 and RBPMS. If you can see in the parentheses there, SMI32 and RBPMS are both done under immunofluorescence staining, and CD8+ is also immunofluorescence staining, and then Ki67 and IBA1 are immunohistochemistry staining. And I'll get into a little later like what these names mean and what they're actually staining for. So the first process, I'll talk a little bit about like how we go from extracting these eyes, extracting the eyes and the optic nerves to finally getting results and getting images of them. So it all starts with cryoembedding, which essentially means that we're using OCT compound to encase the eyes or the optic nerves, or usually the eyes, in this liquid. And then we go to the cryostat, which you can see it's the machine on the right there. And essentially that allows you to take really, really thin slices or sections, usually about 5 micrometers of the eye. And then you can put that onto a microscope slide. And once you have it on a microscope slide, then it's not stained yet. So you have the eye, and you can say, oh, well, it's great, right? No, it still has to be, you still have to do certain things to it so that these certain cells that you're looking for can appear. So that's what immunofluorescence staining is. And in the back, you can see one of the best images that we've gotten of an immunofluorescence stained eye. And the thing with immunofluorescent staining is that you can stain for multiple antibodies. So in the case of the eye, that means we're staining for SMI32 and RBP-MES, which you can see on the bottom there. SMI32 identifies, or it highlights the thickness of, it highlights a region in the retina of the eye, and the thickness of that region is indicative of vision health. And then for RBP-MS, that's identifying the retinal ganglion cells, which are also in that same region. So those also indicate the vision loss if there's less of them. So in that particular image in the back, that's actually the SMI32 staining, but with immunofluorescent staining, you can switch colors. So if we switch to the red microscope, then we will be able to see the RGCs, or retinal ganglion cells. And then another thing that we stain for, this is in the optic nerve, is CD8. So that's present on T cells, which are CD8 plus exhausted, which obviously we're trying to see, are these tumor cells present in the optic nerve? So that's the third thing we stain for, but only in the optic nerve. The second type of staining that we do is immunohistochemistry staining. So this is something I talked about a little bit earlier as well, because it has Ka67 and IBA1 that we're staining for. So again, like after you get your slides off the cryosectioner, you have then you're staining for these things. But the thing with IHC staining is that you can only stain for one cell type at a time. So the first one that we usually do is Ka67, which is a protein that's found in dividing cells. So it can be used to indicate where the tumor cells. So if you have a lot of Ka67 positive cells in your immunohistochemistry staining or image, then you probably have a tumor. And we'd expect with Ka67 that there would be lower proliferation of it in the treated mice. And then secondly, we have IBA1, which is a protein that's found in microglia, and microglia is a macrophage in the brain. So it's generally found in all mice, but we'd expect that it's higher in tumor mice. But the thing is, the treated and the untreated mice are all tumor mice. We wouldn't necessarily expect that the growth of a tumor would change its IBA1 levels or its microglia levels. And then finally, we go into a stage of imaging, and then quantification and analysis, which we use image J. So it's a lot of cell counting and quantification on the computer. So that kind of covers the whole process by which this research has been done by my colleagues and I. I'd like to also highlight that I've worked very closely with Miss Stephanie Bozeman in the lab, and because I don't have that much experience in research, my contributions to looking at protocols in the lab and looking at the lab operation was very helpful, because I can say, does this protocol make sense to someone who doesn't have experience with that? So we've updated the IF and IHC protocols, reorganized them, given them more detail, and then done a variety of, aside from the research that I've done, a variety of like lab organization things with her. So I learned a lot about how can we make a lab function properly as well. Okay, so going back to the research, we've received the results from the anti-TGF-beta treatment at this point in the process. So here we're comparing the CD8 positive T cells proliferation in the IF staining between the IgG control and the anti-TGF-beta treated mice. And you can see on the graph on the right that the anti-TGF-beta treated mice have a very, very statistically significantly less proliferation of CD8 positive cells. And you can see in the two images, we have the one on the left, and the IgG, there's a lot more red cells. You can see those are the T cells. And then on the right, there's a lot less in the treated. And then secondly, looking at IBA1, which we said is microglia, we noticed that there's not a huge difference between the IgG and the anti-TGF-beta. because microglia is found in all these tumor mice at a high proliferation. And then finally, when we look at K67, we see that there is, again, less proliferation of K67, less tumor cells, percentage-wise, in the anti-TGF-beta treated mice versus the IgG control. So here's some frequently asked questions. What future therapies are we exploring within this research focus that I've done under Dr. Barakat? And the answer for that is that this is one of the last stages in the process. The paper has been written largely, and then this is peers getting back to Dr. Barakat and saying, these are some more therapies we want you to explore. So as of right now, we don't really know where that's going to take us. We feel like we've explored a lot of immunotherapies that would contribute to her paper, and we don't know if people come back with more ideas of, oh, you should take this route or you should look at it this way, that would be something to explore, but we don't know. And then looking at the anti-TGF-beta experimental results, what have we learned from those? Might be something that you're wondering. From those results, we learned that TGF-beta is directly contributing to the growth of optic pathway gliomas, but anti-TGF-beta treatment is directly inhibiting that growth, as we can see in the KCC7 levels and also in the CD8 plus T cell levels. And then finally, something else you might be wondering is anti-TGF-beta a viable clinical solution for OPGs? And the answer is no. We're looking at a mouse model. And the thing with mouse models is that they can get really close to reflecting a human version of an optic pathway glioma, but they can never fully represent that. Optic pathway gliomas are not naturally present in mice, and neurofibromatosis is not naturally present in mice. So these are all genetically engineered models, and they cannot fully represent what a human optic pathway glioma may look like. So we can't necessarily define it as being a viable clinical solution at the moment. And then finally, before the end of my presentation, I would like to just talk a little bit about the value of research, given that I've been a high schooler in a lab. I'm always also, like I said, on one of the first slides, I'm a journalist, and I tend to look at things from that perspective. So I'm just going to talk a little bit about what it was like for me being in a lab and what I learned about science and the way science works behind the scenes, which is something we don't really see from the outside world. What does it actually look like to be where all this is happening, where all this sensationalized research is happening? So I'll firstly say that the members of my lab and I, we can surely all appreciate the privilege that it is to work in a renowned laboratory on a world-leading med campus and to interact in like a tight-knit work environment with caring coworkers and mentors, and to do grunt work that doesn't really serve the purpose of money-making, but rather solution-seeking and ailment alleviation. But what also makes working here so special is the core of learning that's deep in the heart of research. Researchers are the students that never graduate out of question asking and problem solving and studying and solution seeking. While it's easy to see research through a lens of the deadlines and the spreadsheets and reports and do-overs that tend to define most jobs, don't lose touch with your appetite for knowledge that drives it all. Secondly, science is imperfect. Science and data, specifically that related to biomedical research, is highly sensationalized in the outside world, as we all know. Take a look at today's politicians. Each points to the conclusions of scientists and researchers to support their every claim and hinges on these words. Or if you Google research laboratory and take a look at the lab coats, pristine benches, and rigid organization in these sugar-coated images. Research is misunderstood to fit into this picture-perfect standard of human achievement, when in reality, when you're really in a lab, you notice that not all protocols are followed to a T, lab coats are not always worn, and experiments are never perfect. But this isn't to say, forget research. In fact, much of the outside trust in research is still very much merited. Despite dealing with chemical uncertainties and puzzling observations, research does remain a high standard of human achievement, and that's because no matter how poorly something goes or how many times something fails, it is always begun again until it reaches a valid completion. However imperfect it is, research deserves credit for relentlessly pursuing the truth to a rigorous standard, simply by weight of the truth's value to the world and its significance for millions. Other industries could learn something from research about getting it right, no matter how deep the failure. And finally, an idea about science that has always bothered me was accentuated as I entered this lab. The idea that the physical world is endlessly complex. First, we learn in school that atoms are the building blocks of the universe, and now we know it's quarks. And in the Gutman lab, reading papers on CAMP, RAS-GTP, KIAA1549, BRAF, and more, it felt as if such research could be done for centuries without reaching any cohesive answer other than more and more naming. It all seemed to contradict what I see as a key tenet of science, one of my favorite quotes from Sir Isaac Newton: "Truth is ever to be found in simplicity and not in the multiplicity and confusion of things." But let's consider briefly the courses of action to take when faced with a world that doesn't seem to manifest simplicity, but instead multiplicity and confusion. Firstly, one delving into such a world may notice it is becoming confusing and stop their search for the truth immediately, scared to violate this key tenet and never to return to the pursuit. Alternatively, one may acknowledge this confusion and simply take it as a fact of life, that perhaps Newton was wrong, and modern scientists simply have to deal with a confusing and disorganized world. Or, and most importantly, one may keep digging into the muck, the confusion, and the multiplicity, but in rebellion of it, holding on to an unwavering faith that deep out of this mud will eventually emerge a simple, shining truth. And that is the route we must take. Thank you all very much for listening to me. I'm sorry I won't be able to answer any questions, but I hope my little FAQ helped a little bit with that. It was really an honor to work in this lab, and it's really an honor to present to all of you and all the research that you all have done. So thank you for this opportunity. Bye-bye.